Hi guys, today we're going to look at the review for Chapter 9, so hopefully this will prepare you for the test for Mendelian genetics. So first we'll go through these questions. Define genetics and heredity. So genetics is our study of the passing of traits from parent to offspring, and heredity is actually how those traits are passed from parent to offspring. So slightly different in their definitions. This is going to be more of a branch of biology, so study of. And this one is how those traits are being passed on. The father of genetics is Mendel, Gregor Mendel. And you should have that in your notes. Think of that as more of just kind of have an understanding of who it was, in case you see his name. Okay, number 2, explain Mendel's process of studying pea plants. And basically what he did, remember he studied pea plants just because they were available. He was an Austrian monk and he worked in the gardens, and so he took the pea plants as his example, and he just wanted to see why there were certain traits that were being passed on and why those didn't ever change. So he started with 2 pure plants for each trait, and in this case, if we use flower color, he had pure purple and pure white, and he called this the parent generation. So that's going to lead us into question 3 here. The parent generation was that first generation that he had, which for him were 2 pure plants for the trait that he was studying. Then he took that parent generation and he cross-pollinated them so that they would trade the pollen from plant to plant. And he ended up with what was called the F1 generation, or first filial generation. So first filial comes from the parent generation. Okay, in the first filial generation for him, he allowed them to self-pollinate, which they would do naturally on their own. And that produced the second filial, or F2 generation. And basically what he had when he started, he had the parent generation being 2 pure plants. So if we used purple and white flower color, he had a purple flower crossed with a white flower. Because they were pure, that means that they were, in this case, big P, crossed with little p, little p. And he had offspring that all showed purple flowers, and they ended up being big P, little p. In the second generation, when he allowed the F1 to self-pollinate, he actually found a different ratio. He found that 75% of them were purple. So in our second generation, 75, sorry, my pen is not working very well, 75% were purple, and then 25% showed the little p trait, or the white trait. Okay, so this 3 to 4 ratio he found, and that white trait that was missing in the first filial generation showed up in the second generation. This is where he led to his laws. And the first law that he came up with was the law of dominance, which said that one trait is dominant over another. So what we use now to show these alleles is a capital letter and a recessive letter. The capital letter for a dominant trait, recessive letter for a lowercase letter for a recessive trait. Okay, and then the law of segregation came from this as well. And basically what that says is that each egg and sperm receive one factor, he called them factors at this point, from each parent. So a parent cannot send both of their letters for that trait on to the child. Instead, they send one of the letters of their trait. So think of the word segregation. It means separate. So we're just separating the trait choices, big letter or little letter, onto the egg or sperm. The law of independent assortment is saying that the factors for the different characteristics are not linked. So they are separated independently, if we look at this word, independently of each other, and they're sorted in egg and sperm independently of each other. Basically, he's saying genes are not linked to each other. Now, there is an exception to that rule we'll talk about later. How are these last 2 laws represented in meiosis? Segregation and independent assortment occur in meiosis when the chromosomes are split into the individual egg and sperm cells. Both occur basically at the same time. They're segregated or separated into the sperm and egg cells, and this is done independently of each other. Okay, our second area in Chapter 9, explain the difference between genotype and phenotype and provide an example. So genotype is looking at the actual genes. So we're looking at the actual letters, like big G, or we can say little g. Okay, phenotype is looking at their physical characteristic. What do they actually look like? So that one's a lot easier. It's also less specific. A genotype is going to be more specific for us when we're looking at something like a Punnett square or a pedigree. Okay. Number 2, explain the difference between homozygous and heterozygous. So homozygous can be one of two things. It can be the same big letters, two big letters like big G, or it's two recessive letters, little g, little g. Heterozygous is two different letters. So in this case, and it's not different as far as the type of letter. I can't get my pen to do that, but it'd be like big G, little g. So homozygous is the same two big letters or two little letters together. Heterozygous is different, big letter, little letter. 3, the terms purebred and hybrid relate to homozygous and heterozygous. So purebred is the same as homozygous, and heterozygous is the same as hybrid. So if you see those terms, that's really what that's relating to. Hybrid, same here. Purebred, same as homozygous. Why are Punnett squares used in genetics? Punnett squares just give us a probability that the offspring will have a certain trait. So we're looking at probability when we do Punnett squares. Now remember with Punnett squares, when we see the results, if there's a 25% chance a child will have a disease or have a trait, it's 25% every time those parents have a child. So it is not like if they already have 3 children with a certain trait, that the 4th one will definitely have that other trait or opposing trait. Okay, number 5, a monohybrid cross, and how is it different from a dihybrid cross? So a monohybrid cross is our typical 4-box cross, and we're looking at one trait. A dihybrid cross is 16 boxes, a 4x4 grid, and we're looking at 2 traits being passed on together, and the probability of those 2 traits being passed on together. All right, number 6, the purpose of a test cross and the possible results of a test cross. So the purpose of a test cross is to look at our chances of your offspring, or sorry, your parent being purebred or heterozygous for a trait. So you would do this if you have a dominant phenotype and you're not really sure if it's a purebred for that dominant phenotype or if it's heterozygous for that phenotype. And so you always, always, always cross in a test cross. You always cross your unknown plant, whatever it might be, with a recessive for that trait. The reason we cross it with a recessive is we know what a recessive genotype is, and we know that that recessive genotype is going to be 2 little letters. The possible results of a test cross: if you have a purebred for your trait, a purebred dominant, all of the offspring are going to show the dominant phenotype. If you have a heterozygous individual that you crossed with your recessive, you'll see some of the recessive phenotype show up in the offspring. Okay, and then here we are with complications and exceptions to the rule. So incomplete dominance, you will need to know the difference between incomplete and codominance. Incomplete dominance is when you blend the traits. So the example we used in class was red flower and white flower, and what will happen is the traits will blend to produce pink. Where codominance is when both traits are dominant. There is no recessive, not necessarily, but there doesn't have to be a recessive. Instead, we have 2 traits that are dominant with each other, and they actually will both show up equally in the offspring. So a codominant trait in humans is blood type. We have blood type A is dominant, blood type B is dominant, blood type AB is codominant, and then blood type O is our recessive choice. Okay, number 3, how are incomplete dominance and codominance different from each other? Kind of mentioned that already, but just make sure we go back over it. Incomplete dominance is a blending of the traits. Codominance, they both show up equally. In class, for the codominants, we use the roan example, the roan hair color for horses. It's red and white hairs showing up in the horse in equal amounts. Number 4, multiple allele traits, and give a human example of a multiple allele trait. So a multiple allele trait means that we have more than just 2 choices in our alleles. So for humans, blood type is also an example for this. We have blood type A. That is one choice of allele. Blood type B, that's another choice. And blood type O, which is a third choice. So multiple alleles instead of just 2 alleles. We have multiple alleles for that. Number 5, define polygenic trait and give one human example. So if we take a look at this with poly means many, and genes. So we have many genes that code for a trait. And they, a good human example is eye color, hair color, and skin color, where there's multiple genes that code for that trait and give us a variety of colors. And then the last one there, define epistasis and provide one example. So in epistasis, this is when the expression of one gene depends on the other gene and what you have for that other gene. So the example we used in class was mouse coat color. Mice can be black coat color or brown coat color, but if they have the no pigment gene, then that trumps the coat color gene. So they're black, brown, or if they have the no pigment gene, they're albino or white. So epistasis just means one gene can trump, or one allele, a set of alleles for a gene, can trump another gene, even if you code for that particular color. Like for mice, if they code for that particular coat color, then if they have the gene for no pigment, they won't show that coat color. All right, that is the end of the review for Chapter 9. If you have any questions, make sure you see me in class.